Okay, so good afternoon, everyone. Welcome to the second lecture of 2022 Overseas Academician Lecture for 120th anniversary of the university. The speaker is our old friend, Professor Chris de Zeeuw. Chris is a very famous cerebral neuroscientist and a member of Dutch Academy of Arts and Science. He is the founder and the chairman of the Department of Neuroscience at Erasmus Medical Center in Rotterdam, and the vice director at the Netherlands Institute for Neuroscience in Amsterdam. He has also been principal coordinator of the European Robotics Program and the president of Neurobasic Mouse and Pharma Phenomics Consortium. Chris has received over 100 grants, including the Pioneer Award from the Netherlands Organization for Health Research and Development, being ranked #1 of all medical scientists in the Netherlands, and the advanced grant from the European Research Council. He has published 3 books on the cerebral function and over 400 papers in many prestige journals, including Science, Nature, Cell, the New England Journal of Medicine, Nature Genetics, Nature Neuroscience, and Neuron, accumulating to more than 22,000 citations. In 2006, he received the Beatrix Award for Brain Research from Her Majesty Queen. And in 2018, he received the International Casella Prize for Physiology. Today, Chris will talk about his study on the cerebellum and exploring the brain based on his research. Chris.
Thank you very much, Yang Ning, for the wonderful introduction, as always. I'm now going to share the screen. You have to allow me to share the screen.
Yeah.
I'm still disabled.
Yeah, you can share the screen.
Yeah, but it says the host disabled the participant screen sharing.
Oh.
If you're the host, you have to click.
I will
and you say multiple. You have to say multiple screen sharing. If you go to the green button,
I will set you at a host. Yeah, you can.
Okay,
yeah.
Great. Yeah. I hope everybody can see me now.
Yeah, great.
Yes, so I chose the title, Studying the Cerebellum to Uncover the Brain, because I received all these questions from Professor Zhu, Yang Ning, and what was clear to me, just putting all the questions together, is that indeed the cerebellum is a very unique structure, because it's very well organized. You have a very clear anatomy. The physiology is very nice because you can recognize all sorts of cellular responses like complex spikes, simple spikes, and you have cell-specific promoters for indeed the Purkinje cells, but also for the interneurons and so on. And you have behavioral paradigms where you can really control the inputs and the output very well. So you can measure the behavior and you can control the inputs. I will give you several examples of that. So I felt you can actually really use the cerebellum to uncover the brain as a whole, because there's all sorts of mechanisms that we have uncovered in the cerebellum and that also apply to the brain. So I will, during my talk, I will just go by all the questions that I received from Professor Zhu, and I ordered them in the following way. I ordered them in 5 main questions, and 3 of them have 2 parts, an A and a B. But here you see all the questions that I received from you, but we will start with question 1. So how to decide whether one is suitable for scientific research or not. And that really has to do mainly, I think, with your desire to do research. You have to be very curious. And when you want to understand things, then you have to be creative to solve these issues. And you have to have then the passion to uncover these things and work hard for it and do whatever it takes to discover these things. So I will give you a personal story. What happened to me after I basically I had my PhD, and then I went back to the wards to work in the clinic. So I'm an MD PhD, but when I was in the wards, I had basically two main offers. One is to go to continue into science and to become a scientist, but there was no warranty whatsoever. You could do some sort of postdoc. And and I already had children, so I had a family to take care of. At the same time, when I was doing the wards in the clinic to become an MD, I also got an offer to become a neurosurgeon in Leiden here in the Netherlands, which was quite unique because in Holland, this very small country, you only have 1 or 2 residencies per year. Now, so I was really thinking about this, what shall I do? I was on the wards, and at that time, at some point during the wards, I was actually allowed for a weekend to go to Barcelona to a cerebellum meeting. And I was going to the meeting, which was a great meeting organized by Linus and Sotelo. And there were all the famous cerebellum scientists were there, but I really couldn't concentrate. So I basically started to walk. I left the building where the scientific meeting was in Barcelona. By the way, there's some green stuff that should be cleaned. I think maybe Ying-Ning, you can clean. Someone's using the cursor.
Sorry, I can't clean. You are the host, sir. You can.
Yeah, okay, maybe I can clean. Let's see now. Maybe I can clean it now. Let's see. No, I think. I can clear. No. Now it should be clear. Yeah. Okay. Yeah.
Okay.
Okay. But so I was walking around in Barcelona with this question in my head. What shall I do? Shall I go stay in science or go in science, or shall I go into the clinic? And then I walked into this museum by Picasso. It was at that time a small Picasso museum. And I just bought a ticket and I went upstairs and I was in this room all by myself. And basically there was only one painting in this room, and that's the painting you're looking at now. It's called Ciencia y Caridad, which means science and care. And for me, of course, this was really resembling my choice. Should I go into science or should I go into care? And as you see on the painting here, in the middle, there's this patient, very white, very sick. And here on this side, there's the medical doctor actually representing Ciencia because he's measuring the pulse. And on the other side, you see the nurse with the child and representing the Caridad, the care. So I was looking at this painting and saying, well, I really like the nurse much better than the doctor. So I probably have to choose for care then, which means I should become then a neurosurgeon rather than a scientist. And while I was thinking this, there was a little bit of rumor in the back. And the rumor came forward. While I was sitting on this couch in front of the painting, and then on the left side, there was someone in a wheelchair coming next to me on the left. And so this person apparently also directly wanted to go to this painting. And then I looked to the left, and who was this person? This was a very famous person, Stephen Hawking. He was then suddenly sitting next to me and also straight looking at that painting. And for me, that sort of thrilled my mind, because for me, he's like the ultimate scientist, theoretical physicist, which I think is a beautiful part of science. So I felt that was a sign, right? So I had to, okay, now I really have to go into science because, he came there. And so I went down. I did not talk to him. I went down, at least not at that time. I bought a little print of the painting. And I went home to my wife and I told her this story. I said, so now I know I have to choose for science. And then my wife said, well, Chris, this is complete bullshit. If I would have been sitting on your couch there in front of the painting, and he would have come suddenly to the left, I would have seen the ultimate patient in him. So I would have become a medical doctor. In that sense, he represented also medical care. And of course, she was right. So apparently it was already in my mind. She said, I already knew you were going to be a scientist. You just had to find out yourself. But by seeing Hawken and seeing the scientist in him rather than the patient, it became clear to me that indeed I should go into science. And this is what I did. And later, actually, 20 years later, I met Stephen Hawking when we actually tried to find a cure for him to get better communication with the world, which we did. And so I met him then later in Cambridge, and I told him this story, and he actually remembered, because I had sent him the postcard to thank him for the guidance that he indirectly gave to me. Why am I telling you this story is that in the end, all of you all will have to find out yourself what is best for you. This is really a very personal choice. Yeah. And you can have all these qualities, but if you don't have the desire to do so, to really sacrifice your life for science, then it's not going to work. You just should feel it from your inside. And if that's the case, then go for it, please. So that's the first and maybe the most important part of this talk. Now we go to the second question. By the way, please feel free to interrupt. I will turn on, or I guess Yingling can tell me if someone wants to ask a question and interrupt me, please do so. Now we go to the second question, which is, scientific research problems often occur at the beginning of a project. So how do we know where to start? What is the most important question? What is a meaningful research question? And this is also a very, very important question. It's also not easy. So I gave a few details and that you can see here. First of all, when you go into a topic, of course, this is usually determined by your PI in the beginning of your scientific life. You have to make sure you read all the literature. And this is more and more of a problem because there is more and more literature every day. Every year, there are millions of papers published. But within your topic, you have to be really the best. You have to know everything, every little paper. And only when you know those, then you can basically determine what is not known yet. So out of everything that's known, you have to find the gap in the market, the hole in the market. And you have to, on top of that, find a hole in the market that is relevant, that you can sort of turn into a global question. Like what, for example, what does depression do with your metabolism, right? Or is the cerebellum involved in cognition? Or in very global questions, if they have not been addressed yet, these are really ideal topics. If your intuition tells you that there might be something there. The question will be even more important if it's generalized. So if you have a question that is not only true, let's say, for the cerebellum, but for the whole brain. Maybe the cerebellum is involved in depression, but maybe a certain part of that mechanism is actually valid for the whole brain. Then it becomes even more interesting. The other part that's really important is how you can answer the question. If you think you can answer the question with a classical technology, that's okay, and that's nice. And then you should definitely do so. But sometimes the questions that you find have not been answered yet because of technical issues. And that is also an opportunity, because if you can develop a new technology that no one has ever done, and yet it might work in the end, then you are shooting sort of two chickens at once, because you will then answer the content question, but you will also have a new technology for which you will also be honored and cited and so on. So the best research question is yes, if it has all these ingredients. So it should not have been done yet, of course. It should be generally applicable, and it should be technically feasible, or it should be possible with the development of a new technique. And when all of this comes together, then you may have something really nice. I'm going to give you one example. Actually, I will give you for this part, the very first project that I was involved in as a PhD. So as I said, in those days, it was much more easy to read all the literature. And basically, when I started my PhD, my supervisor, which was Tom Ruigrok and Jan Voogd, they showed me this screen. He said, Chris, this is basically the brain is organized. So you have the cerebellum, you have the deep nuclei, and then you have here the inferior olive, and they have this sort of a triangular loop that projects from the inferior olive to the cerebellar cortex, through the deep nuclei and back. And then from the deep nuclei, you can innervate the rest of the brain, including the cerebral cortex, but also in the end, the spinal cord and so on. And we want you to uncover how things look like inside the inferior olive at the ultrastructural level. Well, that's a very global question, right? So I thought, well, I really want to know, for example, what kind of neurotransmitters are used here in the projection from the deep nuclei to the inferior olive, as well as from the mesoddiencephalic junction to the inferior olive. And it was already known then by studies from Rodolfo Linas that the inferior olive itself was functionally an interesting story. So, so I felt, you know, given the things that were already shown, for example, there was electrotonic coupling inside in between the olivary cells, it might be interesting to see where these terminals are located. Yeah, so here you see how Linas showed the electrotonic coupling. They just impaled two cells at the same time. And when you inject a hyperpolarizing current or a depolarizing current, in both cases, you can actually get double responses in both cells, even though you change the current in only one of the cells. So I felt, well, let's see what's happening here. Where are the terminals? Because there had been, here you see a gap junction with all the layers. So the cells are really coupled. And you can also see that surrounding the coupled dendritic spines, there are terminals with vesicles around it. And the idea was, based upon all the studies by Linas in the Navanax and Mike Bennett, that you have these coupled dendrites, yes, surrounded by chemical terminals, and that based upon the activity of these terminals, you could get a shunting current so that the two cells, the two dendrites, could become more or less coupled. And because of that, you could maybe control different sets of muscles, let's say in your fingers, as I show here at the bottom. Yeah, this was just an idea, but what first had to be shown is then what kind of terminals are there surrounding. And in Navanax, it was already shown that inhibitory terminals would be particularly, from a functional point of view, suited to mediate this shunting. So I started then to think about a way to show the neurotransmitter with immunocytochemistry and to combine it with anterograde tracing, which had not been done yet. I heard that people were working on it for years already. They tried to combine autoradiography, anterograde tracing of treat-shaded leucine together with pre-embedding immunocytochemistry, but that was very hard. And apparently it didn't work because the grains of the treat-shaded leucine, they spread over the terminal. But I started with a different, I read the literature, so I first detected this question, then I looked at the literature, and I found post-embedding gamma immunocytochemistry, so I made sure I got the antibody, and then indeed I found, here you see two coupled spines coupled by a gap junction, and they are surrounded in this case by GABAergic terminals, as you can see by the black dots, right? And in both cases, there's a clear GABAergic input, so that was very nice. But I still don't know where these GABAergic terminals are derived from. So I want to have this combination, which no one ever did, a combination of anterograde tracing plus immunocytochemistry in the same terminal. And so I thought, well, you know, I can then, rather than using treat-shaded leucine, which takes, you know, half a year to just be exposed in the dark room, I will use HRP, WGA HRP, which is much faster. You can do that in a week. So I start to play and injected treat-shaded of WGA HRP in the cerebellar nuclei, right? And so you see here, you see this reaction product. Yeah, you see the vesicles. So this terminal is indeed derived from the cerebellar nuclei, and it is in the inferior olive because I took the tissue from the olive. Now the question is, then I thought, okay, then I'm just going to do, you know, post-embedding GABA, and then ready, everything will work. And then I see double labeling. And what happened when I did that, as soon as I did the post-embedding GABA, all the HRP was gone. It was dissolved. Initially, I did not really understand it, but I thought about it for actually overnight. You know, sometimes you have to dream about things, and then the answer come in your dream. And what, of course, when you do this post-embedding gamma, you use tritone, which is a soap. So the soap then might be able to solve the HRP reaction product. And then I want to check this idea. So I took serial sections, ones that I treated with post-embedding GABA and the other not, like this one. And then you could see this hypothesis was indeed correct. So this is the serial section. You see the gold dots. So you see this terminal is indeed GABAergic, but you also see there was something here that is dissolved. So if you can see it carefully, there was the HRP reaction product. So no wonder that I could not get really double labeling in a good way in a single section, because it would solve the WGA HRP reaction product. So therefore, I felt, okay, well, this was hard work, but unfortunately it did not work, right? So what do you do with your frustration when things do not really work perfectly? You try to solve them. So I felt, well, maybe what I can do is before I first get this TMB WA HRP reaction product, and then before I go to the post-embedding GABA with tritone, I will first try to stabilize it with a heavy metal. And I look again, I look in the literature, and I found this technology to use DAB cobalt. And this is what I did. So then you know, what you do when the experiment fails, this was one of your questions, try to analyze what went wrong, really try to understand why it didn't work, and look at the details. Yeah, and then once you have insight, try to come up with a solution. And sometimes you have to just take a walk in the park and think about these things. And then if you're lucky, the moment will come. And so at some point then the next day or the next week, I solved the problem by stabilizing the TMB HRP reaction product with DAB cobalt. So cobalt is a heavy metal, and there you go. There it works. So now you see this case, I injected WGHRP in the mesosensor junction. You see here the reaction product, TMB stabilized by DAB cobalt. You can clearly still see the crystal. And here you see the GABAergic terminal. Yeah. And then, of course, it was easy to do the same thing. Now I did it from another area again, the cerebellar nuclei. And here you go. Here you see the reaction product clearly, and you see the GABA, and you see the gap junction. So again, now I could show that all these GABAergic terminals next to the gap junctions were derived actually from the cerebellar nuclei. And I could also then quantify, I could see everything that basically the whole projection from that area, from the lateral cerebellar nucleus and the interposed posteriors, was completely GABAergic. So what I want to, and then you had this pattern, right? So the GABA from the cerebellar nuclei and the mesosensor junction at the excitatory terminals, and together they could manage to control the coupling. So the story tells you that, yeah, things do not always go right, even when you have a good idea in principle, and when you know literature well, you may be trapped into problems which you still have to solve. So try to think about it. But if the general question is good, it will be worth it to think about it and work on it hard. Because, for example, this technology, once we uncovered it and once we developed it, we could also apply it to the rest of the brain. Yeah, so after that, we used it for many other projections. And once you have that in your hands, it's like golden, right? It's easy to score or give a new project to your student. Okay, let's now move to question number 3. This is the question. Probably some of you is interested in molecular genetics, and you think, well, you know, for example, what you say here, you read an article about a gene in what cells and where it's located. It might be involved in the memory formation in Drosophila by altering synaptic function. All this is nice, but why this gene? Why not another gene? Right? Because there's so many genes, tens of thousands of genes. So where to start and which pick should you make? What I feel myself I've been in this situation too, because I also really like molecular neurobiology, is then to look whether the same gene is not just expressed in Drosophila, but whether it's highly conserved among species, particularly in humans, because if it's expressed in humans, then you can also find whether it's involved in human disease. And if a gene is involved in human disease, almost by definition, it means it must have a fundamentally important function. Otherwise, you would not get a genetic disease as a result of that mutation. So I think that is one important point if you can choose between multiple novel genes. And yeah, I would like to show you also an example from my own career yet again. And this is the discovery of the CLIP115 or SELN2 gene, which I did in the 90s, also in the previous century. And I ran into this sort of coincidentally. I was making a cDNA library of the inferior olive. And I was picking up all sorts of new genes. So I thought, wow, this is interesting. This is interesting. You compare it to the library we do this in Europe at the EMBL library at the time. And I found, for example, this gene, and I thought, well, this is great. And we could sequence the whole thing in the old way. I mean, I had to, make all the gels myself, which took weeks and weeks. Now you do it, you just send it in and you have it back the next day. But then to determine whether we were really going to go on with this project or not, we looked at, is this gene also expressed in humans and are there mutations linked to it? And yes, we could see that actually Williams patients, that they had on their chromosome deletions, they had also mutations in which this CLIP gene is involved. And so when we saw that, we decided to go on with this and make a mouse mutant and so on. Actually, we made multiple mouse mutants. At that time, we only made a knockout of the CLIP gene, but now quite recently with friends, Chris de Zeeuw, a former PhD of mine who's now working at the CAS in Shanghai, he made this whole Williams syndrome mouse in which we took out all the genes with the CRISPR-Cas technology. And I will show you a few things which I find interesting and why I really also like this gene. First of all, it's part of many, many genes. And there's a lot of symptoms, including cerebellar symptoms, cognitive symptoms, cranial features, cardiovascular problems, and hypersocial behavior. And I just want to highlight a few because I don't have the time. But here you see that it was binding to plus ends. I could see this under the electron microscope, but I could also see it here very nicely in this preparation that we make together with Spiral of an invertebrate. Yeah, so we can cut the axons and then we can mimic the plus ends of these beautiful plus ends, and you can manipulate the axon and then see what happens, how it regrows, and so on. So it had to be important for development. And this is also what you can see there. Here you see the cerebellum of a patient, a Williams syndrome patient, and you can see actually, if you compare it to normals, that the development of the lobules in the cerebellum is not normal. The same thing we found in the mouse mutants that Chris de Zeeuw made, that the lobulation of the cerebellum is not completely normal, which was mimicking, again, that one of the patients. Here you see Oliver Sacks, who's really also very well known for his affection with Williams syndrome patients because of their feeling for music.
Invited to attend a large meeting of people who share a condition that seems to predispose them to an unusual sociability and friendliness.
They have this typical Williams syndrome face.
Organized for people with a puzzling condition called Williams syndrome. None of them had met me before, but I was immediately struck by the warmth of their welcome. One of them even said, "Don't be shy, Mr. Sacks." And I was soon at my ease.
He's also mentioning the shyness, which they are not.
People.
So what you could see in the face is that the maxilla, the upper jaw here, is a little bit shorter than normal, and therefore the lower lip sticks out a bit and the upper lip is a bit inside. You can see this also in the mouse mutant, actually. It's most likely due to the GTRF1G gene. So here you see the difference. It's a little bit shorter than there, and it's significant. The other one is really the social behavior, which is really fascinating.
Oh, Heidi!
I mean, Oliver Sacks never met this
I
girl.
knew that this was a complex neurological condition, but the little girl who called to me from her front gate seemed perfectly normal.
You?
Me and a few others.
Hello.
What a beautiful orange tree.
How are you? Miss you.
Yet there was something odd in her manner of greeting me. She had the assurance, the poise of a sociable adult. I'd only met her once before, but I was treated like a long-lost friend. A lot of...
Exactly. So even though she didn't know him, she invited him in her room. She was very, familiar as if he was a great uncle that she regularly sees. But this is very typical Williams syndrome patients. So they're sort of hypersocial. And this is also something we see in the mouse. So if you do the free chamber task, yes, you see here on the right that the Williams syndrome mice, they show hypersociability compared to normals, or for sure compared to autistic mice. So I'm just highlighting this to show you that this gene, the CLIP gene, which is involved in some of these symptoms, not all, but they led us to work on Williams syndrome. So if you have to think about, should I work on a particular gene or not, I always think you should try to look at the broader perspective. And if it's involved in human disease, I definitely think you should go ahead with this. If it's not clear, then it might be worth first do something else, because it is a lot of work, this. Good. So now what you should also learn from this is that the interaction between, let's say, fundamental science and the clinic. The way I posed to you the CLIP story, the CELN2 story, is indeed, I started with fundamental science. So I first had this gene, I didn't know what it was doing, and then I decided to work on it because I looked at the websites for mutations from human disease, and I found that it might be relevant. So then I continued to work on it. So I started from fundamental science and worked to the clinic. But you can also do it the other way around. You can also often start with the clinic, because if you have a clinical problem, let's say linked to a particular gene, then you know for sure it also must be really fundamentally important, as I said, right? But now you start from the clinic and then you work your way back to fundamental science. I think most people, when they say, okay, well, yeah, I'm looking at genes because in the end I want to cure people, I want to develop a therapy. For me, it's usually the other way around. Usually I say, okay, well, this interesting disease, and that may really lead me towards a really important fundamental principle in neurobiology, which I think is actually at least as important, probably more important, because that's generally applicable and will hold forever, right? So, and for this, I want to show you the mGlu1 story. And the story is as follows. There was this doctor, Peter Silver-Smith, and he showed me this patient. You see, she is very ataxic. She can barely walk without the help. She can hardly stand still.
Yeah.
So she clearly has a cerebellar problem, right? And so the doctor came to us and said, well, I have no idea what it is. He did all sorts of tests for paraneoplastic syndromes and so on. They couldn't find it. And then I said, well, why don't you give us the serum of these patients from the blood, and we put it on rats and we see what's happening with the cerebellum. And then we found this gorgeous staining here in rodents, you see the molecular layer is completely dark. Yeah. And on top of that, in the granular layer, you saw these unipolar brush cells labeled. And this is almost, we knew it was kind of pathognomic, means sort of unique labeling that you also see when you label mGlu1. If you have a really good antibody for mGlu1, and we recognize that. So, which is also important. It shows that this is not the kind of discovery you can do when you are a student coming in the first week, because you have no idea how many genes there are, what the distributions look like. But if you have looked, let's say, frequently at cerebellar sections, you recognize certain patterns. So we recognized the pattern of mGlu1, and we thought, well, maybe this patient has an antibody against mGlu1. Now, at that time, it wasn't really known yet what how important mGlu1 was. People said, well, it's LTD important, but it's not clear, blah, blah, blah. So we started to look a little bit further. We first want to confirm that it was really mGlu1. So we then put the same antibody on sections of a knockout mouse of mGlu1. So we put this patient's serum here on the wild type on the left, and on the right on the knockout. And you see the knockout is really, clean, right? So it means that the antibody must be directed and against mGlu1. So that was a very nice shot. And so then we continued. We said, well, let's see what's happening if we put it on a slice. And if you put it on the slice, you can see that the spontaneous firing frequency of the Purkinje cells quickly dropping. Yeah. So a few minutes after you put it on the slice, already the simple spike activity is going down, which means it has direct functional impact, not just on LTD or so. Because if you affect LTD, it will never directly affect the firing frequency. Then we said, okay, to make absolutely sure it's the cause, the real cause of the disease, we injected the antibody inside the brain of mice. And here on the left, you see a normal mouse. And here on the right, you see what's happening when you take a normal mouse and then inject it, the same mouse, and when you inject it with the anti-mGlu1, right? So you can clearly see it's very different from before and after the injection. Yeah, so you can see a dramatic impact just by injecting the serum of the patient on top of the cerebellum of mice, of these mice. Here you see how it was diffused. And we did other tests as well, and there was an instant ataxia, and then it was dissolved a few hours later when it was gone again. So this showed that the serum of the patients, and we also purified it for mGlu1, was sufficient to induce ataxia. And this was quite new. So it had nothing to do with learning or LTD. Could be true on top of that, but it had a much more important, much more prominent impact acutely online on the simple spike activity. And therefore, it showed several things, what mGlu1 is doing, but also that you have this active, very acute mechanisms by which you can get ataxia. So the main point of this was that indeed it's very nice to look at molecules and genes, but always try to interpret them in a functional way. And again, the cerebellum is a great structure for this, because you can survive without the cerebellum or by lesioning it, and you can still play and you can measure input output very well. Okay, now question number 4. How to find the right direction in the field of the life sciences, how to maintain a certain sensitivity to scientific research hotspots. Here too, it's very important to keep track with the literature. And in this case, I would say also keep track of the technologies. Yeah, so read review papers in high impact journals, read about new technologies, and try to think how you might apply them to your topic of research as indicated by your PI. Moreover, here too, it's true that the more general functional question you can ask, if it's not answered yet, the more important, the better the chance that you will find something really nice. So try to find a niche, an important niche, driven by both technology and content. I will give you another story about my own career, which was in the last decade, we published in 2017 in Neuron, is the GluR3 story. As you know, MPAR receptors, they're made of different subunits. So you can have GluR1 and 2, but you can also have GluR2 and 3. And GluR1 and 2 already received a lot of attention in the world, in the hippocampus, in the hippocampal field, cerebral cortex, and so on. But GluR3, we knew it was there, but it was not very clear what it was doing. And it was probably partly due because it was very difficult to make good antibodies against GluR3. But we could see in the Allen Brain Atlas that GluR3 was heavily expressed in terms of in situ hybridization in Purkinje cells. So we felt, well, maybe there's something there. And so we started to look what this might be doing. And we started to generate a knockout for GluR3 or import them actually and combine them with other knockouts. So here you see this LTD-LTP experiment. You can see that if you have no GluR1 or GluR3, you can still readily induce LTD. So you get a reduction of the EPSC after combined conjunctive stimulation of climbing fibers and parallel fibers, as indicated here at the top. And if you only give parallel fiber stimulation to induce LTP, you do get great LTP in the wild type as well as in the GluR1 knockout, but not in the GluR3 knockout. So, hey, we thought, well, GluR3 is probably doing something with LTP induction. We looked at the molecular mechanisms, and there's something striking which was different from LTD. As you know, with LTD, the hypothesis is that you sort of internalize the MPAR receptors through endocytosis, which will take, 10 to 30, 50 minutes. And then once it's there, then there's less MPAR receptors in the membrane, and therefore the membrane is less sensitive for incoming parallel fiber activity. So we thought, well, LTP is probably the opposite. This was also the theory. You bring extra MPAR receptors inside the membrane, therefore the membrane would become more sensitive and you would get LTP. This turned out not to be the case. When we played with the molecular pathway by giving forskolin, there was no increase in these spines. Yeah. Whereas when you induce LTD with DHPG, you did see the normal endocytosis inside the spines. So we felt, hey, there must be something really interesting in this mechanism. We didn't understand, of course. And we start to, again, dream and think, and what could it be? And we came up with the idea that maybe something is happening with the current channels that are already there, the MPAR receptors composed of 2 and 3, and that the whole would be increasing. And so we did single patching of single channels. And there you can see that when we stimulated single channels, while you're patching them, you can manipulate them, you can make them bigger. So you can make the hole bigger of the same channel by activating the channel that do contain GluR3. And so this was a new hypothesis. So this is, you see here, the diameter is widening. Yeah. And this is very important. So this could be a new mechanism uncovered in the cerebellum, but also be applicable to other brain regions where it was not discovered yet. And this turned out to be true later. Later, other people showed that this also holds for the hippocampus and so on. But the nice thing, again, about the cerebellum is that you can directly test it in a very well-controllable behavior. And this is actually part 4B. So, yeah, sometimes you just need to do hard work, right? So something you already know pretty much what you can do. And with the GluR3 story, we had to spend a lot of time doing behavioral testing, doing the VOR adaptation, as I will show you, doing simple spike recordings, which takes, a lot of time, like half a year to a year to complete it. And this is basically what you also should be able to do as a scientist. It's very adventurous in the beginning of a project. Let's say you find something and you feel, okay, this is really nice. This is really something I want to do. and you can see that you have something really cool, then you can come into a stage that you have to repeat and get all the numbers. You have to do all the controls. By that time, you sort of should block out almost your creativity and just become a sort of come into an autistic mode and keep repeating the experiment that is important, just to make absolutely sure that everything is correct that you're saying. So you have to, as a scientist, you have to also be able to play with different modes of your brain. And it's not always easy for everybody, I see. So what we did in here is we subjected the glue-free knockouts, cell-specific knockouts and global knockouts both to the VOR adaptation. Some of you have already seen this video for other talks. So you put the mouse in the center of the turntable and you record the eye movements with a camera. And you first put the animal in the dark while measuring the eyes. You compare the size of the pupil and the movement of the pupil with regard to the corneal reflection. Then you turn on the visual image and you get an out-of-phase stimulation. Yeah, so you get increase in retinal slip. And then you do this for like half an hour. Then you put the animal back in the dark, and then you will see that the VOR gain, the amplitude of the normal VOR in the dark is increased after the increase in of stimulation. Yeah, so I will show you what the results are. Here you see the knockout. You see there's hardly any learning. In the wild type, there's huge learning. And all of this can be linked to this new cellular mechanism. And then you have to, if it's really LTP, then you expect that an increase in simple spikes is the cause of this, right? So you first do normal recordings and you see indeed an increase during the learning of the simple spike activity when the gain goes up. Yeah, and then if to know whether this is sufficient, you do optogenetics. You stimulate, and dependent on the intensity of the stimulation of the Purkinje cells, you can also see that again, the amplitude of the eye movement can be increased, just to make the whole story round. But all of this is hard work and not always easy and takes months and months. And you basically have to shut off your deep thinking, and you know, you will not have a brilliant idea every day, and you don't want to because you just need to get this paper out, right? Whereas in the beginning, everything is very exciting and yeah, but that's a life of a scientist. Sometimes it's boring, but it needs to be done. And sometimes it's very exciting. But in the end of the day, you want to uncover something that has not been uncovered before. And that's really important. So then I think it's really worth it to go through all these stages. Now we go into the last questions, which are really, I think these are two commercial questions. One is really about direct commercializations, where I promote my students to commercialize their findings. And the second one is, what about big data, and how are these two related? And I will try to tackle both questions from you by showing you what we have done by developing Erasmus BV, and particularly Blink Lab. In Erasmus BV, we started to basically build blink boxes by doing eye blink conditioning in mice. And we are, we have been working on this for like 20 years. If you want to do eye blink conditioning, we can provide you with great eye blink boxes for, I think, relatively little money. It's not a profit company. There's no, in Erasmus BV, we don't make any profit. We only develop products and we want to help other scientists. There's not a real profit company. The other company, Blink Lab, which is the follow-up, that's using eye blink conditioning in humans to help patients. That company is really supposed to make real profits, and we have high expectations of this. And this is what I will focus on now. This was basically developed by Henk-Jan Boele and Bas Koekoek, two of my PhD students. And I'm also involved. We just started it last year, and we developed an app by which you can do eye blink conditioning. You can do trace conditioning, you can do pre-pulse inhibition, you can do all sorts of tests. And I will show you a few examples. So why did we develop it? Well, one of the main reasons was that autism is a huge problem. So here you see the numbers of the US. We think by the year 2025, so that's just 3 years from now, about 500 billion per year will be spent on treating autism in the US alone. Yeah, and the problem is that there is no real early diagnosis at this moment. So people often, when they're young, like 3, 4, 5 years old, parents already feel something is wrong, but they don't know what, and then they do complicated tasks, which can last a whole day, and then at the end of the day or a week, then when the child is almost 5, then the doctor will say, okay, yeah, your child probably is suffering from autistic spectrum disorder. If you would have known this earlier, let's say when the child would have been 2, you could have done appropriate interventions that would have helped the child, and you would have saved a lot of problems later on. Now, so it's very important to have early diagnosis, and we felt we can do this with the app that we developed. And so we developed the app. We are trying now to test it in in children with autism spectrum disorder. Here you see that we really want to make it an earlier stage, and we want to find, let's say, behavioral biomarkers in the app results. So in the old days, we had these children coming in the clinic. Children are uneasy, of course. They get this huge helmet on the head, and they get puffs on the eye and so on, and it's all very cumbersome, and the recordings are not that great. So why then not develop an app that you can have your smartphone, that you can have in your hand? You can do it at home. Everything can be automated, and all the data can stream into the platform that we built. So it's better, it's faster, it's cheaper, and it's more pleasant. And again, it can be used by people in academic institutes, by clinicians, but also by parents at home. You can already download it through the App Store. And yes, so again, just to give a brief history, in the old days, already more than 150 years ago, people started by looking at reflex modulation. Pavlov, of course, in the early 1900s, started with eye blink conditioning, differential conditioning. Hundreds of papers have been published, including by us, finding the neuronal mechanisms. And it wasn't really until very recently that actually we had the idea of smartphones already more than 15 years ago, but the computer technology was not in place yet. But in 2012, then by using AI and machine learning to analyze paradigms, things have become much, much more available, and to bring the two things, the science and technology together. Yeah, so that's what we did. And I show you one example now. This is Stephanie. This is pre-pulse inhibition. So if you give just a pulse, you get this reaction. So just a small sound. But if you give a pre-pulse, yeah, then you see that the pre-pulse will actually prevent the big output, the big pulse, the big reaction in the second stage. And this is a normal reaction, right? But if you then do the same thing with an autistic child, for example, here, the autistic child will also have a big response following the big pulse. But if you do pre-pulse inhibition, you see that there is no inhibition. So the pre-pulse does not generate an inhibition, unlike in the control subject. So here you see the difference between the control subject and the autistic subject. So pre-pulse is having a clear impact in the control, but not in the autistic child. You can compare it to other diseases like ADHD, which have also the same reaction to a normal pulse, but there you see that the pre-pulse, they're hypersensitive, right? So the pre-pulse does a different thing compared to autistic children and controls. So you can have a lot of sort of signatures of different kinds of mental disorders, and you can also actually evaluate the treatment. So you all know for ADHD, you can use Ritalin. Yeah, so this is also methylphenidate, as you know. And we can measure this very quickly. In practice, in the daily practice of a psychiatrist, they usually ask patients to use it for several weeks, and then they have to come back and fill in the questionnaire, and then they guess whether it worked or not. But we can actually measure it. So we, just by doing PPI, for example, just looking at the pre-pulse, we can measure from hour to hour what the impact is of Ritalin. Yeah, so it's also in that regard, it's, I think, a very valuable evaluation mark. Now, we are now bringing it on the market. The app is fully developed. Everybody can download it. You basically only have to watch a movie. And while you're in the movie, you're being subjected to all sorts of stimuli. And you don't know what's really going on, but it's completely impossible for you to sort of fake a certain behavior or not. And we're now bringing it, making it FDA approved, and then we go on from there. It's privacy protected, of course. And we do not only take the eyelids, we take the whole facial expression. Yes, this also has to do with big data, because we want to know, we want to compare recordings, let's say, of people in China, of people in Australia, in South America, North America, Europe, and so on. So we really need big data as well. But of course, neuroscience is not just big data, it's everything. You need to have big data, you need to have hypothesis-driven research, you need to have, deep thinking and dedicated experiments and so on that are highly complicated. This is just to show to you that we are really already having Blink Lab in almost all parts of the world. And yeah, we have the team is expanding almost every week, and we hope to really go on the market soon. So we expect to be profitable in a few years from now. So far, we're only investing, but it looks like this valorization product may yield some profit. Okay, now I think I did still well within an hour. And so this was my presentation.
Thanks, Chris. Great talk. So, students, if you have any questions, you may directly ask Chris or type your questions, both in English or in Chinese, will be okay. So, Chris, I have a question. So,
Go ahead.
Yeah, I know actually I never know the Williams syndrome has the hypersocial activity. So does this dysfunction also depends on the cerebellum or other brain structures?
Yeah,
Quite different with the autism. Yeah.
Yes, the hypersocial behavior of Williams syndrome patients is probably both cerebellum and extracerebellum. The reason is that you can just say if you lesion particular genes, for example, only in Bergman glia in the cerebellum, then you see only part of the social deficits, and probably it has to do with interaction also with, for example, the amygdala and the cerebral cortex.
Okay. Xiaoyang, you have a question? I know you open your phone.
Okay. Oh yeah, so I see a question here in the chat box. It says, what about your suggestions? Is the research work, can it be scooped? Yeah, that's an interesting question. So let me give you one example. Actually, I'm gonna share again my screen. Okay, let me see. I'm gonna go here. Just one, I will give you one example. I have to go really back to the beginning, sorry. This was very fascinating because I found in a publication, it's really, I cannot, Yeah, so this figure, right? This is a figure from my thesis, from my own thesis. And I never published this figure in a publication anywhere myself. But at some point, I read a paper by people from Japan. And they published this figure with the concept. And they did not even cite me. They did not cite my thesis. They did not cite the work on which this was based. And so I thought that was quite something. So yeah, scooping that happens. I wrote them, of course, a letter. Said, well, that's really coincident. That is exactly the same figure, and they apologize. But yeah, these things happen. So I don't know what you really want to know, but I guess you want to know how to deal with it, right? And it's very difficult. You have to, I was always extremely open. I always said everything, but I've been scooped several times. And yeah, sometimes, you know, good ideas occur in multiple places, right? In general, I would say nowadays that you have to be a bit careful. When you have to be, you know, when you're on something really nice, it's better not to present it yet at meetings. Also, for example, the Gordon meeting is known. You should tell something novel, but I've been really scooped at Gordon meetings with the exact same data. So people are supposed not to use it, but it just happens. It's just so tempting. So I became a little bit more careful over the years. What is your view, Yang Ning?
Yeah, I think so. I agree with you. Yeah.
Yeah.
Yeah, I agree with you.
You want
so
to be open,
difficult.
but it's
Yeah.
yeah. How to choose an appropriate PC supervisor. Okay, that's also a very good question. I think, and I'm sort of let's say I have a pretty big lab still, but I would not go to a big lab. So you should not go to my lab. The reason is that when I did my PhD and my postdoc, I was working in small labs and I got all the attention from the PI that I wanted. So whenever I had a small question, I could go there, always available. If people want to see me now, you know, I often have to give lectures or I have to go to some management issue or whatever. So if I were a PhD, a starting PhD or starting postdoc, I would be happy to be in a small group. Now, on the other hand, if you are a more advanced postdoc and you want to have a lot of freedom and do your own thing and you can do it, then it can actually be good to be in a big lab if you get all the freedom and also all the money that you need to do your work. Then it might be a little bit different. But your question is, as a PhD supervisor, I would go to a supervisor who is still having a small lab. But of course, you also have to see signs that this young supervisor with a small lab is upcoming and is doing really well. So if they have a few really good and nice papers, but the lab is still small, that might be a nice combination. Okay.
Okay, any other questions?
I don't see any question further.
Yeah. So if you have no questions, so that's thanks Chris again for his great talk, especially today his story in doing the science, combine his story and to answer to answer the questions of our students about how to deal with his scientific careers. So thanks, Chris.
Thank you, Yang Ning. And we stay in touch about the project, right? So that's nice.
Yeah.
I'm impressed by the FMOS work. I really like it.
Yeah. I think I wish to prepare the manuscript in the 2 weeks. and then I will
Okay.
send you.
There's still one
Thanks.
more question. There's still one more question. They say,
Okay.
how can undergraduates quickly find articles that are worth reading? Yeah, I would still start with reviews. I would just take Nature reviews and go from there, the most recent ones. Okay.
Okay.
Okay. Bye-bye. See you later.
Thanks.
Bye-bye. Bye-bye.
Bye.